Natural killer cells and HIV pathogens. What are they? Natural killer cells, NK cells, are lymphocytes in the same family as T cells and B cells. They are the body's mechanisms of defense against foreign invaders. They are best known for killing virally infected cells and detecting and controlling early signs of cancer. They were named natural killer cells because they do not require activation to kill cells that are missing self markers of MHC1 class 1. These are the most aggressive of all lymphocytes and have no immunological memory. They are part of your innate immune system, aka non-specific immunity. What types of cells are they? NK cells belong to a group of innate lymphoid cells. They are not phagocytic and are one of the three kinds of cells differentiated from the common lymphoid progenitor. They are known to differentiate and mature in the bone marrow, lymph nodes, spleen, tonsils, and thymus. What do they do? The role of an NK cell is analogous to that of cytotoxic T cells in the vertebrate adaptive immune response. NK cells provide a rapid responses to virus-infected cells attacking at around 3 days after infection and respond to tumor formation. NK cells are different from typical immune cells because they have the ability to recognize and kill stressed cells in the absence of antibodies and MHC1. In contrast to natural killer cells or natural killer T cells, NK cells do not express T cell antigen receptors or pan-T markers CD3 on surface of IgB cell receptors. They patrol your blood and lymph looking for abnormal cells and are unique in the aspect that they can kill a person's own cells if they have become infected with a virus or have become cancerous. They kill in contact by binding to its target. When a cell becomes infected, they stop making major histocompatibility complex on their surface. When they detect this, they do not swallow, such as phagocytosis. Instead, they attack it. They bind to the infected cell and poke it with an enzyme that triggers holes on its membranes, known as apoptosis, programs cell death. After a natural killer cell has destroyed an organism, it displays pieces of the pathogen on its outer layer. They make up about 5 to 15% of lymphocyte population. What is a pathogen? A pathogen is anything that can produce an infect or disease. This term is used to describe an infectious microorganism or agent. The specific study of a microorganism includes microscopic pathogen organisms. It is called microbiology, while the study of the disease that may include these pathogens is called pathology. HIV. What is it? The human immunodeficiency virus, aka HIV. There are two species of viruses that infect humans. Over time, they cause acquired immunodeficiency disorder, or AIDS. In most cases, HIV is sexually transmitted by, or is it sexually transmitted infection, also known as STD, and occurs by contact or transfer of sexual fluids or blood. Non-sexual transmission can occur from an infected mother to her infant during pregnancy, nursing, or childbirth. HIV infects vital cells in the human immune system, such as helper T cells, macrophages, and dendritic cells. HIV infects vital cells in the human immune system. It attacks helper T cells, macrophage, and dendritic cells, which affect the humoral response in the immune system. The outer envelope of HIV is covered by glycoproteins, which mutate frequently, thus making it hard for helper T cells to detect them. Once attached to a specific receptor on the T cells, it begins to fuse with the membranes together, where it then releases 2 RNA strands and 3 essential replicating enzymes that begin to treat it as its own and make more copies of it inside the cell. After this, it then leaves the cells and matures, infecting more and more T cells. NK cells and HIV research. NK cells play a critical role in vital immunity. Specifically, NK cells directly mediate immune pressure, leading to virus evolution. NK cells, receptors, genotypic profiles, clonal, um, and functional capacities have all been implicated in the virus containment. Specifically, NK cells directly mediate immune pressure, leading to virus evolution. Natural killer cells function in abnormal, thus allowing HIV to multiply inside a human cell. HIV is hard to treat because it mutates constantly at a rate so high. More than 10 billion virions are created each day. When the T cell falls below 200, or when the count falls below 200 per cubic millimeters of blood, it then becomes known as AIDS. In conclusion, we should all take microbiology and pathology serious, especially in the light of recent events concerning COVID-19. HIV and AIDS research continues to be necessary, or to be a necessary endeavor until a cure is found.